Good morning, everyone. Today, I want to share a story of survival, evolution, and the remarkable power of genetics. A story that unfolds on a tiny island in the Galapagos archipelago and centers on one of the most iconic examples of evolution in action, Darwin's finches. Charles Darwin once proposed that when similar species compete for limited resources, they may evolve divergent traits to reduce that competition. This idea, now called ecological character displacement, plays a central role in how scientists understand both community structure and speciation, the formation of new species. But for a long time, scientists struggled to find conclusive, real-world evidence of this happening. That changed on a small island called Daphne Major. Let's zoom in on two finch species living there: the medium ground finch, Geospiza fortis, and the large ground finch, Geospiza magnirostrus. In 2004-2005, these birds endured a severe drought. Food became scarce, and competition for hard seeds intensified. The larger ground finches, with their powerful beaks, had the upper hand. Medium ground finches that had evolved larger beaks, perhaps once advantageous, were now at a disadvantage. Why? They were too weak to compete with the larger species and too inefficient to process smaller seeds. As a result, many died. But those with smaller beaks survived. This was character displacement in action, natural selection favoring finches whose beak size diverged from their larger competitors. And unlike many hypothetical models, this event left clear, measurable impacts on the finch population. But here's where it gets even more fascinating. Scientists wanted to know, what genetic factors were behind this survival pattern? Why did some birds thrive while others didn't? Using genomic analysis, researchers discovered something extraordinary. They identified a region on the genome, a single locus roughly 525 kilobases in size, that showed strong genetic differentiation among large, medium, and small finch species. At the center of this region is a gene called HMGA2. HMGA2 is known in other animals, including humans, for influencing body size. For example, in mice, a mutation in this gene leads to a pygmy phenotype, and in humans, variations in HMGA2 affect height and craniofacial dimensions. In Darwin's finches, researchers found that this locus had a major impact on beak size, not beak shape, that's controlled by another gene called ALX1, but the overall size of the beak, which directly affected survival during the drought. When scientists looked at the genotypes of medium ground finches that lived or died in 2004-2005, the pattern was clear. Birds with two copies of the S allele, associated with small beaks, had the highest survival rates. Birds with two copies of the L allele, associated with large beaks, were much more likely to die. Heterozygotes, those with one of each allele, had intermediate survival. This pattern suggests that the HMGA2 locus has an additive genetic effect on survival. In fact, a single SNP, just one base pair difference near HMGA2, was found to explain 27% of the variation in beak size among medium ground finches. That's an enormous effect for one genetic variant. And it gets better. This same locus explained 30% of the evolutionary shift in beak size that occurred during the drought. So, what does this tell us? First, it shows that a single gene, HMGA2, can have outsized influence in an evolutionary context. This gene became a genetic focal point during a moment of intense environmental stress, driving a measurable shift in the population. Second, this story reminds us that evolution doesn't just work slowly over millennia. It can act rapidly, within a few seasons, when selection pressure is strong and genetic variation is already present. And third, this study gives us a clearer picture of how genetic architecture can shape the path of adaptive radiation. Darwin's finches famously diversified into multiple species adapted to different ecological niches. The fact that HMGA2 variants existed across species and were shaped by both selection and introgressive hybridization, gene flow between species, suggests that these finches have a shared genetic toolkit that evolution reshuffles as needed. Interestingly, the S allele at HMGA2 appears to have come from the small ground finch, a different species that occasionally interbreeds with the medium ground finch. This kind of gene flow, called adaptive introgression, may have played a key role in helping the population respond to the drought. It's a striking example of how evolution can be a collaborative process across species boundaries. Now, I want to end with a broader reflection. This research doesn't just deepen our understanding of Darwin's finches. It challenges a long-standing assumption that evolutionary change is always driven by many genes with small effects. Here we see that sometimes a single gene, when aligned with the right ecological conditions, can shape the fate of a population. In this case, a drought on a tiny island turned a genetic variant into a survival ticket. That's evolution in action. And it's not just about beaks. It's about how life adapts, endures, and transforms in the face of change. Thank you.